Welcome back to BioP 105 Intro to Biotechnology. And today we're going to talk about microbiology and cell culture methods and talk about some of the things that we're going to be doing in lab with different kinds of bacteria. So, in this chapter, we're going to be talking about the structure of basically bacteria. So, we're going to really focus on prokaryotes, but we'll also mention eukaryotes in contrast. We're going to talk about the major groups of prokaryotes and how we identify them with a technique called the Gram stain. And then we'll also talk about the uses of bacteria and to some degree, the eukaryotic cells that we'll talk about in biotechnology. So first off, cell biology is the study of cells, and microbiology is the study of microorganisms. Microbes or microorganisms, as they're also called, are too small to be seen with the naked eye, so we need a microscope. And so the groups that we include are the bacteria, and that's the one we're going to focus mostly on today, the fungi, which also include the yeast, which we use for baking and for fermentation, but also the algae, the protozoa, and viruses, which are below the limits of the microscopes that we have, but you can see them in the electron microscope. So we know that bacteria were really the first cellular life forms on Earth, and we know they've existed for over 3.5 billion years. So really, the origins of us, really, I mean, our oldest ancestors are dating back to the bacteria. So we are surrounded by microbes throughout our environment. They're on our skin and our mouths, in our digestive tract. We know they perform many important functions in educating our immune system and helping us digest our food. And there's approximately 10 times as many bacterial cells in and on the human body as compared to our own cells. Now, our own cells are bigger, so by volume, we have more human cells. But in terms of just cell number, we're actually more microbial than human, I guess you could think of that way. If you look over the planet, we can see that microbes are adapted to some wide ranges of conditions. You can virtually find microbes anywhere. So you can find them in the polar ice caps, you can find them on glaciers, you can find them in deserts and hot boiling springs, and even deep in the ocean in hydrothermal vents. They are actually some of the primary producers in that environment. So some very unusual microbes there. So just for comparison, this is a typical animal cell here. So you can see the nucleus in blue, the cell membrane, and then of course the mitochondria and all of the endomembrane system where we produce proteins and internal compartments. So by contrast, then here is the typical bacterium. So it's usually on the order of 1/10 or less the size and much smaller volume than a eukaryotic cell. Their connection to biotechnology is that in sort of traditional biotechnology, microbes were, first of all, the old-fashioned way used to make food, but we use them today in recombinant DNA because their genetics are very well characterized. We know a lot about them, and we can manipulate their chromosomes. So it's something we can actually do in our lab pretty easily. So some of the areas of research ongoing are producing a variety of materials for treating diseases, but we also use some of these recombinant organisms to produce biofuels. And then there's a whole field of synthetic biology, which is trying to re-engineer the genome of an organism to have a desired function. And often this is looking at ways to maybe clean up pollution or other ways to produce fuels that we don't have right now. What's interesting is that we estimate we've really studied less than 1% of all bacterial species. And so we really have a lot of work to do in terms of identifying them and studying them. A lot of it has to do with the fact that we just haven't cultured everything. So who knows what's out there in terms of, things that could be produced or discovered that could provide some benefit to us. So we didn't even know about microorganisms until the 1700s, and Leeuwenhoek is known to have produced or made the first microscope and actually recorded his observations. So he recorded bacteria and protozoa in a variety of environments from his own body and things he collected in the environment. But it wasn't until at least 100 years or more later that the connection was made between specific microbes and disease. And that was a pretty important breakthrough. So Robert Koch was a German physician, and he was working on a disease called anthrax, which is known to kill cattle. It can also be deadly to people as well. So he was able to culture the bacteria in a lab he actually set up in his own house. And this organism was the organism Bacillus anthracis. And the disease that it causes is the disease anthrax. So the name of the organism is Bacillus anthracis, and the disease that results is anthrax. So he developed a series of steps that he later termed Cook's postulates, and we now know as germ theory, which connects specific microbes to specific diseases. And the steps are that you observe the microbe in all cases of the disease, but you don't observe the microbe in a healthy individual. So in this example, with a rabbit, healthy rabbits would not have this bacterium, and the animals that develop the signs and symptoms of disease, you can find the bacteria. So you can see the bacteria here, and this would be something that you would see in the microscope. Then the next step, so first step is that observation. Then the next step is to actually culture the microbe. And so towards the end of this section, I'll be talking about culture methods that we're going to do in the lab. So you need to have agar plates or some kind of culture system where you can grow the organism. And when we say a pure culture, we mean that we just have one species present. And that's a really important requirement to get to that organism to study. So now that you have the organism growing in the lab, then you have to test and see whether it actually does induce disease. So you would take that culture and inject it into a healthy animal. And in this case, it does cause a lethal infection. So you would observe the same signs and symptoms. So whether or not all diseases cause death, or organisms cause death, but in this case, that would happen, but you would see the same signs and symptoms that you'd seen in your original experimental animals, study animals. So step 3 then is, so the step 2 is the culture, and the step 3 is then the lab animal step. Then the 4th step, and I think this is a really important one, where he makes a full circle and doesn't make an assumption that the animal died. He actually proves that the animal died of that infection by recovering the same organism, and he recultures it. So that recovery and then reculture of the same organism is a really important way to sort of close that loop. So when I say be able to list and describe Cook's postulates, this is what I want you to be able to do, is say, you first off have made that observation and connection of a specific description of the disease, and you've seen the organism in the tissues and so forth. You get a pure culture with one species, you do your experiment in your lab animal, and then you reculture the same organism from your experimental animal. So I think it's obvious that this is a really important series of steps to connect a microbe with disease, but you can also see problems with this where you couldn't accomplish all of the steps in Cook's postulates. So for instance, what if you can't culture the organism? And there's cases where we know we can't culture it. We can do the other steps maybe by passaging it through animals. What if you can't actually infect an animal with it? Like maybe humans are the only host. So now you're going to have to observe this, but you're not going to be able to do the experiment. You're just going to make lots of observations that every time you see this organism in a patient, this is the specific organism that causes that disease. There are ways around this now that we can do recombinant DNA in animals. You can often make a model of the disease in either through adapting the pathogen to the microbe, to the pathogen, or to the host, or you can actually humanize. You can put a human receptor for a virus or something like that into a mouse. So there's ways to adapt and make your animal model experimentally, but you can see there could be more required. And then, of course, you can't do the experiment. So ethically, you can't actually put a microbe into a human subject if it's a dangerous pathogen. You might be able to do it with some of the milder cold viruses or things like that. But clearly, there's many cases where you couldn't do this. So there's a lot of situations where you can't perform all the steps, but we can still make most of the steps and make that connection. So once we discovered that microbes cause disease, it was important then to think about ways to treat disease. And so the development of in vitro methods for looking for agents that could possibly curtail the growth of microbes became very important. And so Alexander Fleming in the 1920s made an accidental observation where he left some plates with bacteria, and this is the bacteria that's Staphylococcus aureus that causes skin infections. He got some fungus growing on the plates, and if you look carefully, there's sort of a zone here where he's got some inhibition. And so he was able to identify this compound that was being produced, and it was later then further identified through the work of Florey and Chain, and they were able then to bring that to industrial production of this antibiotic by massive growth of the fungus, basically. And that was the first scale up of a drug to specifically treat bacterial infection. So penicillin was a huge breakthrough, and that led then to the quest or desire to look for other types of antibiotics from other microorganisms. So I guess we can ask the question, are bacteria our friend or our foe? We know there are many very important things that bacteria do. They fix the nitrogen, so they convert atmospheric nitrogen, or N2, which is held by a triple bond and very difficult to break triple bonds. Bacteria are able to break that bond and then convert the nitrogen into a form that plants can use, and that then becomes the protein in plants, which we either eat directly or we eat the animals that ate the plants. So nitrogen fixation is a real important part of getting nitrogen into the food chain. We also know on the other end of the scale, when we have waste materials, a variety of bacteria and fungi help break those down and convert them to simpler compounds. So we'd have a lot more waste than we already do if we didn't have microbes. We use microbes to produce food. So you think about the yogurt that you ate, possibly, that sour, tangy taste is from the activity of lactobacillus in the yogurt. And also sourdough bread is through the activity of a combination of the bacteria, but also the yeast that are helping to produce the CO2 that cause the bread to rise. And then, of course, I just mentioned we get a lot of antibiotics from actually other bacteria as well as fungi. But we know they cause disease. So you can say kind of both sides, there's good guys and bad guys in the group. So turning our attention to the structure of microbes, we'll talk first about the major groups. So we have, if you look at overall, of all cellular things on the planet, we have three major, what we call domains. And you might have learned the 5-kingdom system of classification, but the domains are kind of over that, on top of that. So the big domain that we're familiar with is the domain Eukarya, which are all eukaryotic microbes or organisms. So in the microbial world, they include the yeast, the algae, and the protozoa, but also in this group, not microorganisms, we'd also include the plants and the animals. Then prokaryotes are actually two domains. They're one group that we're familiar with, the bacteria, but this other group, the archaea, are very interesting because they're found in very unusual or extreme environments. So archaea share properties of both eukaryotes and prokaryotes. So evolutionarily, they're sort of a middle branch, I guess, in terms of cellular structure. So a lot of archaea live in very extreme environments, like very hot. They live in the hot springs of Yellowstone. They live deep in the ocean. They live in very salty environments. So there's something called halophiles because they actually require salt to live. So the Great Salt Lake and so forth would be a good place. And then the ones that live in hot environments are called thermophiles or hyperthermophiles, depending. So this is a representation of the three domains. The bacteria, we'll talk about a variety of groups, the gram-positives and the gram-negatives. I won't talk as much about the archaea, although I will mention some important ones that have contributed to biotechnology, and I think there's a lot still to be discovered in this group. And then the eukarya are the things we're more familiar with, which include the plants and animals and so forth. And I always like to point this out on the tree of life. Who's our closest relative? If you're looking at all the animals and the next closest group, it's actually the fungi. So fungal organisms have many similarities to us. And that's something most people probably don't think about. So we're going to stop at this point and we'll continue on in the next section looking at structure of bacteria and how we can identify bacteria with the gram stain.